This in vitro human alveolar model uses relevant immune cells as a powerful tool to predict the acute immune response of substances, including nanomaterials and novel drugs. This method combines human lung epithelial cells and human primary immune cells to assess donor-specific reactions. The use of fresh and frozen monocytes provides flexibility for the experimental planning.
Numerous studies have shown that this model can provide an insight into the intracellular communication between epithelial cells and immune cells. As this demonstration includes all the important details for assembling the model, a person trained in cell culture could easily follow the written and visual instructions to replicate the experiments. Several steps in the blood isolation protocol are complex and require special handling. Therefore, they are demonstrated with all the required details that they can be easily followed.
For the isolation of peripheral blood monocytes from human buffy coats, split the Harvest Buffy Coat bag contents evenly between two 50 milliliter conical tubes. Carefully bring the total volume in each tube to 50 milliliters with PBS, and mix the tubes with 3 gentle inversions. Then split the Buffy Coat PBS solution equally between 4 new 50 milliliter tubes. Then add 10 microliters of CD14 positive magnetic beads per 10 to the 7th total cells to the tube, and mix well by pipetting. Upon filling, detach the pipette from the pipetter and immediately plug the upper opening of the pipette with a thumb. Then place the filled pipette at the bottom of one tube.